So guys, we have seen that how the degradation of glycerophospholipids takes place. Now let's see today that how the degradation of a second important phospholipid, that is sphingophospholipid, takes place. As an example, we will take the example of sphingomyelin, a very important phospholipid. So if you see here, as I have told you that a phospholipid, specifically here is the example is the sphingomyelin. It's composed of a ceramide moiety and phosphoryl choline moiety. The ceramide moiety in turn is composed of sphingosine moiety and a fatty acid. So first of all, an enzyme known as sphingomyelin, sphingomyelinase, it will cleave the bond between the phosphorylcholine and the ceramide moiety. So as a result, the phosphorylcholine will be separated and you will end up with the ceramide moiety, which is composed of sphingosine and the fatty acyl group, or fatty acid group rather. You know that sphingomyelinase is an lysosomal enzyme. As I've told you, it removes the phosphorylcholine group and we get the ceramide moiety. So then what happened that another enzyme, ceramidase, it will cleave the bond between the fatty acid group and the sphingosine group, and as a result of this, the fatty acids and the sphingosine will be separated. And then these fatty acids and sphingosine then perform their own functions. The fatty acid, of course, will be involved in the energy production, while the sphingosine is involved in different signaling pathways. Also keep in mind that ceramide and sphingosine also involved in the activation of a very important signaling pathway, that is the protein kinase C based signaling pathway, which involve the phosphorylation of many protein and initiate many important signaling pathways. One of the most important manifestation of these signaling pathways is the apoptosis, or the cell death. So you have seen that phospholipids are, though they are not very complicated molecules, but still in terms of their importance, they are highly significant molecules. And also you should know that it's not that any molecule which is involved in energy production that is important for any metabolic pathways, can also do lots of other function, as we have seen in the case of phospholipids. Now let's ponder on, or I mean, I need you to really meditate and think about our topic a little bit in more, with more seriousness, in more detail, so that you can answer the questions which I will ask you now. This is, our favorite topic or favorite section, that is the let us ponder. So first question: what is the benefit of amphiphilic nature of the phospholipid? What benefit it provides to phospholipid? I mean, we have been saying that most of the lipids, being most of the time, they are hydrophobic, or major portion of them is hydrophobic, but here we see there is a hydrophobic and hydrophilic portion also. So what is the, in case of phospholipid, so what is the benefit of, what is the significance of phosphate group? I mean, because of this one phosphate group, you get a whole branch of compounds, of lipid compounds. So what is significant? What phosphate provided? Why not calcium? Why not sulfur? Why not sodium? What is so important, so significant in phosphorus, so special in phosphorus, that it gives a whole, I mean, change the whole characteristics of compounds? What is the significance of sphingosine moiety? Yes, I have told you, you have learned that phospholipids are mainly structural or functional compounds. They are not mainly used for production of energy. So what makes them, I mean, what is the factor behind it? What is the cause of it that they are not mainly the energy producing compound? Or take it like that, what makes a compound a mainly energy producing compound?